Hello, grade 11 students. In this video, we're going to talk about the structure and the chemical composition of the DNA for grade 11. First of all, what is the DNA? DNA, where it's located and in what form? Deoxyribonucleic acid, DNA, is the molecule that contains the biological instructions that make every one of you unique. For example, Mahdi is a unique individual, Fran is a unique individual, Maya is a unique individual, Lean is a unique individual. All of you, Maya is a unique individual. Every one of us is a unique individual due to this DNA. DNA is passed from adult organisms to their offspring during the process of reproduction. So your DNA is passed from your parents, from your dad, and from your mom to you. That's why you are very special and you are unique. Because the cell is very small and the organism they have many DNA into the cell, each DNA must be tightly packed. It must be tightly packed. This packaged form of the DNA is called the chromosome. So the chromosome represents the tightly packed structure or the tightly packed form of the DNA. It's called the chromosome, this X-shaped structure. The cell is very small and the organisms they have many DNA molecules per the cell. So each DNA molecule must be tightly packed. You have to pack them and pack them tightly, and this is the form of DNA which is called the chromosome. DNA is made of chemical building blocks. They are called the nucleotides. As you know, these building blocks they are made of 3 parts. We have the phosphate group, the sugar group, and one of 4 types of nitrogen bases. To form a strand of DNA, nucleotides are linked into the chain. This chain will be along with the phosphate group and the sugar group and remaining. So we have both sugar group and we have phosphate group for forming the strand of the DNA. So this is the DNA strand, double stranded. You can read it 3 prime to the 5 prime. This is double stranded, where the nitrogenous bases or the nucleotide we have A adenine, T thymine, G guanine, and C cytosine. And we have the base pairing between these two where C and G they are complementary, A and T they are complementary. So as you see here, adenine and thymine they are complementary. The red, sorry, with the yellow they are complementary with each other, as you see here, and the blue with the green they are complementary. And then we have the sugar phosphate backbone or sugar phosphate bond or groups. The 4 types are nitrogenous bases are found in the nucleotide. They are adenine with the A, thymine with the T, guanine with the G, and C, which is the cytosine. The order or the sequence of these bases determines what biological instructions are contained in the strand. For example, the sequence having ATC GTT might instruct for the blue eyes, while ATC GCT might instruct for the brown. So you see here, so one change in the nucleotide sequence, ATC, ATC, the same. G, G, T, it's C. So one change in the nucleotide sequence from T to C, it changed the eye's color from blue to brown. Each DNA sequence that contains instructions to make a protein is known as a gene. The scientists they use the term double helix. Why? Double helix because it has this shape. This is called a double helix structure. So they use this term in order to describe the structure and the two-stranded chemical structure of the DNA. It's called double helix. This is the double helix structure. Okay? Also, the shape is like a twisted ladder. It's like a ladder, but it's twisted. It gives the DNA the power to pass along biological instructions with great precision. It's very precise. This is the structure of DNA where C and G they are complementary, A and T they are complementary, where we have hydrogen bond between these two bases, A and T they are bonded together. This is called the hydrogen bond, and we have the sugar phosphate backbone between the two strands. Homologous chromosomes and the homologous chromosomes and their sister chromosomes. So the point of the intersection of these two chromatid, they are called the centromere. You have the short arm, it's called the P arm, long arm, which is called the Q arm. These two chromatids, they are called the sister chromatids. So we have here the non-sister chromatids upon replication, they become sister chromatids where every one of these chromatids is going to be duplicated. Forms of genetic information carriers. We have chromatin and we have chromosomes. Chromatin is during the interphase, while chromosome during the cell divisions. So we have during the interphase we have chromatin, while during the cell division we have them in the form of chromosomes. Here, the most important application during this video, you have to pay attention to this. This pneumococcus is a bacterium that causes pneumonia in mammals. It exists as two types. Pay attention to this exercise or for this application. Pneumococcus is the bacterium that causes pneumonia in the mammals. It exists as two types. We have type S. It's a virulent strain. It's surrounded by a capsule. Upon the injection of mice with this strain, pneumonia was observed and the death of the mice was followed. So S stands for smooth. So type S, whenever it's injected, it causes pneumonia and finally it causes the death. Meaning that type S is fatal, it can cause the death. While type R, it stands for rough, is a non-virulent that has no capsule. So I repeat, we have two types. Type S is a virulent strain. It's surrounded by a capsule, and upon the injection of type S into the mouse, we have pneumonia, and finally they die. So as I said, S stands for smooth, while type R is non-virulent, it lacks a capsule, and R stands for rough. In 1944, Avery and his team, they performed again this experiment. The results are performed below, and they show us two strains of bacteria, they have different features. When we inject them with type S, smooth one, we inject them in the bacteria into the mouse died. While injection of type R, the mouse survived. Document B, the experiment of Avery and his team, the bacterial transformation. What did they do? They have the S. They get the chemical extraction of the S. They get only here they put the ADN, it's the DNA of the S. And they get the R protein. So they mix the R and the S. S is known to be what? S is the virulent that causes the death, while R is the non-virulent that lacks a capsule. So they mix the S DNA, the DNA of the smooth strain, and they mix with the R, which is the rough, which is the alive one, and they inject it into the mouth. The result was the death. So what does this mean? This means that the DNA of the S converted the R into S. So R with some S derived from the transformation, DNA of the S plus R, they transform the R rough strain and the non-virulent that doesn't cause death into an S type that can cause death. This means that the genetic information they are located where? Inside the DNA. The DNA is the carrier of the genetic information. And interpret these experiments. What is interpret? Interpret, you have to analyze plus indication. After injection of the type S strain of bacterium pneumococcus into mice, the mice they died. So what does this indicate? This indicates that type S is fetal. While after the injection of type R strain of bacterium pneumococcus into the mice will remain alive. What does this indicate? This indicates that type R is not fatal, it doesn't cause death. So you have to use this word from now on. You don't have to say it doesn't cause death. You have to say fatal or not fatal. Okay? Document B, after the injection of the DNA of type S strain and the live strain R into the mice, they die. S, we said that is the virulent, it causes death, while R is the non-virulent that lacks the capsule. This indicates that the extracted DNA of the S strain, the DNA that was extracted from the S strain, transformed the R into the virulent S. So, and this will cause the death. So the DNA of the S, of the smooth type, transformed the R into S, into the fetal type, and this will cause the death of the mice. So what can we conclude? That the DNA is the carrier of the genetic information and not the protein, which is in this case, which is the R strain or the alive R strain. So from this, we can know that DNA is the carrier of the genetic information and not the proteins. So here we identify the different forms. We identify the structure of DNA, where it's located inside the nucleus, and it's transmitted from the generation, from the parent to their offspring, genetic reproduction. You have to know that the package form of the DNA is called the chromosome. You have to know that the nitrogenous bases, they are A, T, C, and G. The A and T, they are complementary, while C and G, they are complementary. And you have to know that the DNA is connected through sugar phosphate backbone, and we have the nucleotides, we have the hydrogen bond between these nitrogenous bases like A and T, we have between them the nitrogen bond. Also, you have to know that the form of the genetic information carriers, they are chromatin during interphase, and they are chromosomes during the cellular division. Also, we identified the experiment that's done by Avery and their colleague that the type S is a virulent strain. Whenever it's injected into bacteria, it causes the death, while type R is non-virulent. It doesn't cause the death of the mice. Whenever we inject DNA from the S and we inject the R alone, the DNA of the S converted the R into S, and this will cause the death. This indicates that DNA is the carrier of the genetic information, where the DNA of S transformed the R strain into the virulent S strain.